Hello, everybody. I'm here to talk to you today about soil biology. This is Willie Durham. I'm the regional soil health specialist for with the National NRCS Soil Health Division. A little bit of my background: I was raised in South Texas, 95,000 acre ranch. Though I was involved in animal science and range management, I chose to go into production agriculture. Graduated from Texas A&M with a BS, Bachelor of Science degree in entomology and a master's degree in agronomy and agricultural chemistry, actually. And I started my career as a chemical fertilizer field man. And I went on to be a regional agronomist for a major seed company working in Texas, Oklahoma, and Tamaulipas, Mexico. I joined NRCS in 2002 as a conservation agronomist. Worked my way up to resource team leader in Uvalde, Texas, and then on to state agronomist here in Texas in 2009 up until 2015 when I joined the National NRCS Soil Health Division as a regional soil health specialist, where I currently cover the states of Texas, Oklahoma, Louisiana, Arkansas, Mississippi, and New Mexico. So a big hello to everybody from Texas, and I'll get into the soil biology module. All being said, I'm going to speak to you today about soil biology. From the previous presentation on soil health basics, you learned that soil health is the capacity of the soil to function as a vital living ecosystem. That function is provided to us by microorganisms in that soil. And in fact, 90% of soil function is mediated by soil microbes, which leads to the enhancement of four key ecosystem processes in every agroecosystem, whether it be cropland, pasture, range. Those processes include the carbon cycle, okay, which is an energy cycle. Actually, it feeds and fuels the soil system. Plants photosynthesize. They build biomass to be decomposed by microorganisms, and they also put root exudates into the soil, which feed many microbes performing other critical functions within and around the plant root system, from things like mycorrhizal fungi that you may have heard of to free-fixing nitrogen bacteria and also phosphorus-solubilizing bacteria that all are around that particular root system. They all perform functions in that soil system. The second ecosystem process is the biocommunity cycle, which of course is the soil food web, the biology, and then plants, animals, okay? It's all the biodiversity that is in that system, and that biology that's there, it's there to create structure and porosity to that soil. In turn, you get a good functioning, what we call a good functioning water cycle. You know, you get good infiltration, you get availability of that water in the soil. As pore space and aggregates and things are built, you're better able to capture rainfall, for instance, or irrigation. And it's also, you're able to hold on to water and helps you prevent much of the evaporation that can happen during a hot summer. So once we get that good functioning water cycle, then the fourth ecosystem process is the nutrient cycle. Okay, this is where we have soluble and plant available nutrients available to you, and you're getting good retention and recycling of those nutrients because you've got a proper water cycle. But you have to understand that 85 to 90% of plant nutrient acquisition within these soil systems, okay, is microbially mediated. Microbes are a big plus in the soil health management system, and that's why this basic understanding of soil biology is so important to soil health. Okay, so we have to understand that the biological soil function is actually ecological management and the management of those four key ecosystem processes. What I'm going to do here is I'm going to use a beer analogy. Okay, that's right, that's a beer, B-E-E-R analogy in order to explain or to link soil biology to soil health. So essentially, as I use this beer analogy, I gained it from John Stickett and Jay Fuhrer in North Dakota, and it helped me learn how to link biology to soil health. And so the situation that we have is that we're working with a bunch of beer lovers who continue to ask that question: can I have more one more drink of beer, one more inorganic shot of nitrogen or phosphorus, one more irrigation, one more rainfall event. Okay, they're always needing these particular things, and so you're in this room with a beer lover who wants that last drink, wants that added shot of fertilizer, or irrigation, or rainfall. So here's what you do. Move to the next slide. Okay, so here's what you do. You give him or her two options to select from. Now, it's rather difficult with a virtual presentation, but you give them two options. Option one, you set 4 cases of cold beer down on the table, which he's welcome or her is welcome to drink to satisfy their short-term needs. Okay, or option two, you set down the equipment and the ingredients needed for one to make all the beer they could need in a lifetime, plus you give them a refrigerator unit to keep it cold. Okay, and guess what? It's all powered by sunlight. Okay, so my question to y'all in the audience, and you can just tell me your answer, but with those two options, when I walk out of that room, which option is the best for long term for a system that will last, into the future? Do you satisfy your short-term needs, destroy soil function degradation to the ecosystem processes, or do you build what we consider to be a beer dynasty, which is actually an analogy for a soil health management system that will lead, and last a lifetime? And I might add, it's not as input intensive. So because you're bringing back all those ecosystem processes, which lessen the need for outside inputs and to replace those lost ecosystem services and functions. So that's why the discussion of soil biology is so important. So which option is better? Option one for short term, or option two for long term sustainability?
Option two.
Yeah. Actually, the majority is option two. We have to understand that this has to be a long-term, sustainable type of system. Okay, so when we go through this biology module today, just to point out, our objectives today are going to be to list one key activity performed by each of the 3 functional groups for soil organisms. We're going to list 2 organisms that represent each functional group, and then we're going to describe biological hotspots and how they relate to key ecosystem functions. Okay. So we also refer to these as the spheres of influence. So what we're doing is we're building habitat for biology, okay, in these soil systems. Okay. As you go through your PowerPoint, as we go through this particular PowerPoint presentation, and I know you're working with a particular class worksheet that is asking you particular questions, but as we go through the soil biology module, think about the 5 biological hotspots and think about your scenario in your breakout session, okay? And then answer these particular questions, okay? Identify which hotspots you think are most limiting, which organism you think will be negative impacted, okay? And then also, is there a specific soil health principle or manipulation approach you think could help change that biology to improve soil health and function in your system? In other words, which particular management practices may be applied and how are they to be applied in your particular scenario, okay? And just simply describe which one might be best, okay? So just keep that in the back of your mind as we go through the soil module, biology module. Okay, soils host a vast numbers and mass and diversity of microorganisms. In 1 hectare or about 2.5 acres of soil, it contains about 15 tons of organisms, which is equivalent to the weight of about 20 cows on 2.5 acres. Okay, that's primarily, it simply means that we have about 3.3 pounds per 1.2 square yards of soil. Okay, so in that acre of healthy soil, it can harbor up to about 27,000 pounds of biomass. There can be 10,000 pounds of fungi, 6 to 10,000 pounds of bacteria, 2,700 pounds of algae, 1,800 pounds of earthworms, 600 pounds of protozoa and nematodes. So just know the soils host 25% of the Earth's biodiversity, and greater than 90% of that has not even been cultured or fully identified yet. Okay, so we're still learning about the soil system. These plant, microbe, animal interactions are involved in all soil processes and functions. And remember the fact, as mentioned along with that definition earlier about soil health, as to be in the continued capacity of the soil to function as a vital living ecosystem, that 90% of that soil function is mediated by these soil microbes in that soil. And that's why the soil biology module and understanding the soil microbes is so important to soil health. Slide. So what you see here, this is a simplified soil food web. Historically, soil biodiversity was studied by mapping soil food webs like you see here. This approach detailed the chain of energy transfer in the soil, and it's based on grouping organisms according to their trophic role and food preferences. Okay, so in a very simplistic manner, the food web is fueled by plants and photosynthetic bacteria that fix carbon from the atmosphere through photosynthesis. Okay, so plants provide the residues, they put that photosynthate, those root exudates into that particular soil. Other organisms then obtain their energy by breaking down the plant residues and organic compounds, or by consuming other organisms. Okay, and those represent the various trophic levels like you see here. And during decomposition and consumption, nutrients are converted from organic to inorganic forms, and they're made available to plants and other soil organisms that are there. So there's a lot of pros to this understanding. It's very useful for understanding nutrient cycling and energy flows as they convert those organic nutrients into inorganic nutrient forms, or possibly they could be organic forms that are available to plants, like amino acids, such as, is an example. But they provide the foundation to study what we consider to be functional soil biodiversity. Okay, so we're going to get right into it here. One of the things that we're going to go over is that we're going to classify the soil organisms into 3 broad functional groups. Okay, the soil biological community controls all soil processes and functions important to plant productivity and multiple ecosystem services. Soil organisms also have co-evolved for hundreds of millions of years, and they interact in positive, mutualistic, and negative ways, where you have things like predator versus prey relationships. Okay, but conserving biodiversity also translates to conservation of important functions or services that the biological community provides you in that soil system. So keeping all that in mind, soil biodiversity and soil functions are complex interactions between both inherent soil properties, things like climate, management, and the life within that soil. A simplistic view is to categorize the soil organisms, like you see here, into the following broad functional groups. And these include the soil ecosystem engineers that you see here up at the top, the biological regulators, and the chemical engineers, aka microbial decomposers. So the following slides that I'm going to go into here will explain what these groups are primarily responsible for, key representatives that will be identified, and a brief overview of specific functions, and where they are found in soils and their abundance will be provided. Okay, so when you look at this particular slide and this illustration, the arrows that are crossing among and between the different groups are an indication okay, that the groups are not exclusive to certain members, and multiple members can be influential on other functions, okay? But in order to kind of follow some structure and help us understand key groups, we're going to go through them by identifying these dominant players, okay, in these three functional groups: the ecosystem engineers, biological regulators, and then the biochemical, engineers, okay? So the first one that we're going to go over is, of course, the ecosystem engineers. And these are the larger organisms such as earthworms, ants, termites, as well as plant roots that modify or create their own habitat. And in doing so, they create biopores, as an example, that channel air and water into that soil. They mix organic materials during burrowing, and they help build resistant soil aggregates. Okay, so by regulating resources and redistributing and regulating access to resources, these organisms create hotspots, okay, their own biological habitat that support high numbers of these microorganisms. Okay, they support themselves. So a large source that fuels the entire soil system and impacts this soil system are plant roots. Okay, that's why we have to have continuous living roots as much as possible year round. And this is the first group of those ecosystem engineers that I'm going to discuss here. Plant roots can cause physical weathering as they grow and they expand inside of cracks, say in rocks. Roots and decaying vegetation also produce organic compounds such as solvents, acids, or alkalines that enhance the actions of percolating rainwater, for instance. The two main types of root systems are fibrous and taproot. Fibrous roots are the traditional structures, formed by primary and secondary roots branching out in all directions in the soil. And by contrast, we have taproots that are characterized by a single firm root growing straight down with minor roots developing on either side. So other specialized roots also do exist. For example, the tuberous roots of sweet potato are modified for the storage of nutrients and water. And so the number of known plant species has been estimated to be around, say, 400,000. Okay, so just know that ecosystem engineers, they redistribute soil particles, microbes, and organic matter. And they literally, what they're doing is they're preparing much of what the next groups will process. So just know what some of those representative members are, like earthworms and millipedes and centipedes. Know that they build that pore and network and aggregate structure to the soil. Very important that you grasp that. Okay, now chemical processors, also known as biochemical engineers, which involve soil microbes, which include things like bacteria, fungi, protozoa. They regulate 90% of the energy flow in the soil, and they are responsible for decomposition of plant organic matter into nutrients that are available for plants. Okay, so soil microbes are also responsible for stimulating plant growth, plant protection, and the production of multiple antibiotics, which are used for human and animal health. Okay, so I just mentioned to you, we normally in our virtual trainings, we have a poll question right here, but it's also listed on the slide. And this is virtual. We tend to ask, and this is one of the key things to understand about chemical processors. And that is which soil functional group is responsible for 90% of the energy flow in the soil. Okay, and actually, it's that function is regulated by these chemical processors. Okay, they regulate 90% of the energy flow in the soil, build the soil organic matter and the aggregates that are there. So you literally have to have the biology to accomplish this goal. Okay. Next slide here. The next group that we're going to look at are the biological regulators. Okay. Biological regulators include small invertebrates such as protozoa, nematodes, potworms, springtails, and mites. And through grazing, predation, or parasitism, these particular organisms regulate populations of other invertebrates and microbes. Okay, so this particular functional group, just know that they regulate the other populations of soil organisms that are in that soil. Okay, we have a lot of them that are there that really work for us and do a lot of good things for us, like protozoa and nematodes. These things are not all bad. They're beneficial. They help us change those organic nutrients into inorganic nutrients in that system. What we have here is, so what I'm going to go into now is understand that optimal activity in most ag systems occurs when conditions are just right. Okay, there are numerous factors that influence biodiversity and activity of soil organisms. Although microbes can exist in the most extreme environments possible, most agricultural soils benefit from the following conditions. You need to have near neutral pH. You need to have moderate temperatures. You need to have soil water at field capacity, good infiltration going on, good aeration occurring, have a low bulk density, have abundant and diverse food sources, diverse soil pore sizes for the different types of microorganisms. And then we have to have minimal amounts of contaminants or salts, that can be a real issue. So essentially what it equates to, it really equates to what we consider to be the Goldilocks principle, okay, where things have to be just right for these microorganisms to manifest themselves more or less in the soil. And so one of the other things that we have to also understand is that the majority of life in the soil is not active in an active state, but often remains in a resting stage that allows them to stay alive when conditions are not conducive, but they rebound very quickly when conditions improve. An example of that is that greater than 90% bacteria in the soil are actually inactive because they have not been able to move, say, toward a organic substrate to use, but they spring back to life very quickly whenever you re-wet the soil to redistribute soil organic carbon and things that these microbes need in that soil. So if we want to encourage a diverse and healthy habitat for the soil organisms, we must provide food, nutrients, and proper aeration, pH, water. In other words, the conditions to support their particular habitat, okay, or their existence in that soil system. Okay, so it's very important to understand that. So also understand, because of what I just mentioned to you, that much microbial activity is actually seasonal. Microbes are impacted by temperature and moisture, and the greatest activity, as you see here in the slide, is in temperate systems, occurs in the late spring, okay, or early summer. And this is when temperatures and moisture are optimal for growth of these microorganisms. So not all organisms are active at a particular time. Some remain dormant only to become active later in the season, okay? So this results in benefits kind of being stretched out over time, as we grow that particular crop, as we go from, germination all the way up to harvest, okay? So that's one of the things that we want to grasp and we want to understand. And to give you an idea of how habitat is built, I want to show you something here. This is called bioturbation. If I can get this slide to activate. It's the mixing of plant residues into soils and sediments by biotic activity. Okay, and it's one of the fundamental processes in ecology. As decomposition occurs, habitat for other microfauna is created, along with increases in gas and water flow through that particular soil. And here you can see a system without soil fauna on the left and with soil fauna on the right. and you can see various soil fauna such as earthworms, potworms, springtails, mites, and isopods this is happening over about a 15-week period. So during this period, whereas only small fungal activity can be seen on the left, the layer in the right side is almost completely incorporated into the soil due to the interaction between those microbes, microfauna, and mesofauna. Okay, so this is a particularly good video. If you want to have that for presentation or to show to any producers, you can. This video is part of the Soil Life and Action Project, and the movie can be used for educational purposes.
So last night we came out to the farm here and we set some pitfall traps. And a pitfall trap is a way that we can identify or assess the numbers of soil-dwelling insects that kind of travel across the surface of the soil. And to do that, we use some pretty simple tools. We used a bulb planter, so to use this to excavate the soil, and then we put some cups into the soil. So we wanted to dig that hole about as deep as these cups were, so that when we go out to check them, we can take the first cup out and have anything that we've collected in that first cup, but that second cup would remain in the hole. So if you want to do longer term monitoring, you could then put another cup back in there to then continue to collect any insects that travel across the surface.
So we irrigated last night, so it was important to have this little roof on it. You can see the water on it. So we just used 3 twigs and a plate like this to keep it covered. And so then when we pull that off, then here's our pitfall trap down below. We did put a little soapy water in it so that if we had any flying insects come in, they can't just hit the bottom of a dry cup and fly out. So soapy water kind of keeps them in place so that we'll get to see them the next day.
And if you notice, it was seated well within, right at the soil level or a little bit below, because we want those beetles to walk across the soil surface and basically hit that opening and fall right in there. So let's put it in here and kind of see what we have.
A few springtails, a lot of the little ones coming across. You'll see a lot of the little springtails, which are important at, decomposing organic matter and things. But what's left then?
So I pulled out here, there's quite a few ants, and I can try to sort them out a little bit here. So I'm going to kind of put some ants over here. Oh, we actually have a nice looking centipede.
One thing that is good, especially since a lot of these organisms are really small, is bring your magnifying glass or your hand lens. We also sometimes put a proscope or a magnifier on our phone to snap closer pictures too, to maybe help you in identifying the organisms a little later, because a lot of them are small, especially the little springtails and mites and thrips and things like that get pretty small. So that gives us a pretty good idea of just after one night what kind of population. There's spiders I see there that were crawling around in all of this cover, in the darkness of the night. They're all hungry. A lot of these are predators. The centipede's a predator. The spiders, of course, are predators, and they would be eating what potentially might be pests, because this is getting ready to go to a crop. So we want that population, we want that habitat for the beneficials to be here well ahead of planting the crop, so that they can do their work before we actually plant a crop here.
Now we're going to move into the biological hotspots. And despite the fact that most microbes are in their resting phase, there are biological hotspots where population size and activity are orders of magnitude higher relative to the bulk soil. And so a highly functioning soil will support all of these particular hotspots, and they're sometimes referred to as spheres of influence. Okay, these hotspots include an area immediately surrounding the roots that's known as the rhizosphere or the root zone. Okay, which is in very common terms is just known as the root zone, everything around the root. Okay, where the soil microbes are stimulated by the release of sugars from plant roots as an output of photosynthesis. Okay, and then as the plants produce above ground biomass and they die, you're going to get this. These residues are going to form a litter layer. That's known as, in this particular paper, that's known as the detritosphere. Okay, so when you get that those above ground residues, they form that litter layer. Those things are then, those residues are decomposed, okay, by not only microorganisms but also earthworm activity. Okay, and then you also have those earthworms putting tunnels and stuff in that soil system, and you have root channels also. All those support increased biological activity, and they aid in the formation of macropores and strong, stable aggregates. Those earthworm tunnels and root channels, that's the area known as the drillosphere. Okay, so that's a very important process. So these hotspots support multiple processes that are working in concert together. You know, as we're doing all this, breaking down the organic matter, putting roots in the ground, we're also doing processes such as creating aggregates in that soil. And we're creating more aggregated surfaces within that soil system. That can be very important to nutrient cycling, okay, as an example. Okay, and you're also creating, as you create aggregates in the soil, you create pore space. Okay, so when you create those pore spaces, that's known as the porosphere. So essentially what you have here is that we have what are known as biological hotspots. And just to let you know, there are 5 biological hotspots. Okay, that's the root zone or rhizosphere. Then you have the detritosphere or the litter layer. And then you have the earthworm and root channel, okay, known as drillosphere. And then you have aggregate surfaces or aggregatosphere, okay? And then you have what we term to be a pore space, which is the porosphere, okay? And actually the porosphere is the lungs and circulatory system of that soil system. So these are vast areas of influence that are resulting from biological activities through ecological succession, okay? So just kind of get an idea of what we're talking about there. Okay. Okay, so our idea here is that we want to capture solar energy, and soil organisms are involved in all aspects of how that soil functions, and these functions include the nutrient cycling that I mentioned to you, soil aggregation, plant protection and productivity, detoxification, maybe chelating toxic metals, okay, which helps us out. Also, you get organic matter formation within the soil, okay, which is I'm going to discuss in more detail in the next slide. But just know and understand we're capturing solar energy, we're making organic carbon, photosynthate, we're feeding those sugars and that photosynthate to microbes, and those microbes are performing certain functions within that soil, okay? So that's that first biological hotspot that I mentioned to you, and that's that rhizosphere and all the microorganisms that are involved in that, okay? So this next slide, what I'm going to show you here is that, you know, when we build all that residue and stuff up on the top of the soil surface, you know, a lot of people think that that's where the majority of our organic matter comes from, but actually very little comes from there. And just note that the reason that it takes so long for organic matter to accumulate many times in soil from crop residue or cover crops or manure on the surface is due in part to microbial respiration. Okay, and most of that is going to be returned to the atmosphere as carbon dioxide. What we do know now is that this function that you see here called the microbial carbon pump is that we know now that photosynthesis and plant root exits are now recognized as constituting the primary pathway for soil building. Okay, this is an anabolic process. I've included this because we really need to understand the difference between what's happening when it concerns the carbon cycle in the upper surface of the soil, and then what's happening in the soil with association with proper habitat like mycorrhizal fungi, free-fixing nitrogen bacteria, phosphorus solubilizing. These types of microorganisms are actually building organic matter in that soil. They're humifying. This is where the humic substances and stuff are produced. The biology is doing that for us. And we also have to understand that as we create that porous and aggregated soil, this is the habitat for free-fixing nitrogen bacteria and phosphorus-solubilizing bacteria. And they are within those particular aggregates that are produced in that soil. So plant roots inputs, build soil carbon 5 times faster than carbon from above ground biomass. Okay, so this is critical to the soil health management system. This is why we have to have live living plants in these particular biological hotspots. Okay, so just know that whether you're producing, grain or fruit or vegetables or hay or, whatever it is, you're first and foremost, you're a light farmer. You really have to understand the importance of capturing that sunlight and solar energy to make organic carbon and photosynthate to fuel this whole soil system. So build your photosynthetic capacity, have live living plants out there, enhance that photosynthetic rate, feed those microorganisms to build soil organic matter in that soil. Okay, so that's very, very critically important. And I mentioned to you about, these microorganisms that you're bringing this habitat, into, and this next particular slide illustrates that. These are known as rhizosheets. So whenever you see a plant that's putting out photosynthate or sugars to form a biological activity with microorganisms, you're going to see that soil sticking to the roots. Okay, that is actually creating the habitat. That right there is known as that rhizosphere. That's that area right there around the roots. You're creating what are called rhizosheets. These are healthy rhizosheets on a cereal, 8-week-old cereal rye that was seeded in early fall. And I might mention that this was an out fertilizer. Okay, so the plants are stimulating the microbes to create those things such as fixation of nitrogen, solubilization of phosphorus, those types of things in the soil. Okay, so very important to understand that what's going on. Okay, and just know that this hotspot for chemical processors, that's where we have all these root exodus and chemical signals that stimulate all these particular microbes and predators. We have a lot of symbiosis going on, protection, chemical signaling, nutrients, and resilience all happening within that soil system. And just know, particularly these plant roots, that's what we start out with. They help anchor that particular plant. They keep that plant in the soil. They help prevent soil erosion. It holds the stem upright. We also get absorption of water and dissolved nutrients that are in that water. This is an area of storage for starch and other nutrients. And then, of course, we have habitat that provides the nutrients to the microbes that helps with further aggregation into that soil. Okay, so the idea is we have to continually do this year in and year out, okay, stimulate these particular processes. Now, within the root zone, we have the rhizosphere. We have these key microorganisms. We have bacteria. These tend to be the most numerous. I'm not going to read off everything in the slide, but 2 to 5% of soil organic matter. They propose 2 to 5% of soil organic matter, but they're responsible for 90% of the energy flow. A gram can contain 10 million bacteria, 1 million species. And they can comprise, 1/2 to 3 tons per acre in that soil system. When it comes to the fungi, we have saprophytic, we have mycorrhizae that are symbiotic, we have pathogenic forms of fungi, like your seedling diseases, and then we have up to 5 tons of these microorganisms in the soil, as far as fungi are concerned. And then we also have the protozoa and nematodes. Remember, those are the ones that regulate. They consume microbes and they recycle nutrients in these particular soil systems. So all these are very, very important. In conventional tillage systems, the microbial diversity is reduced due to the changing environmental conditions and disturbance. So when you have conventional tillage, you have more bacteria and protozoa, especially ciliates, because these organisms can tolerate compacted soils, anaerobic condition, and reduced pore space. So our idea is to do no-till. And so we want to use no-till and cover crop system. We want to reintroduce that to these agricultural systems and know that the bacterial levels do not necessarily decline, But actually what happens is that we get an increase in fungi, nematodes, and other larger protozoa in those particular systems. They will repopulate the soil, and they will increase the soil microbial diversity within that system, creating more of a balanced system, okay, than what we had previously when we left it fallow, barren fallow, maybe for 6 to 8 months between crops. Okay. But one of the key microorganisms that I've already mentioned to you is actually mycorrhizal fungi. And, these, it comes from a Greek word meaning fungus root, okay? Brassicas are an example of a family that is non-mycorrhizal. However, most crops are mycorrhizal, including things like onions, corn, cotton, wheat, soybeans, potatoes, alfalfa, many of the other tree crops, all the way up to coffee and even rubber. So just know that these microorganisms are there. They actually comprise, slide to work here. The plants use about 5 to 20% of carbon from photosynthesis to feed fungi. Fungi increase absorptive root surface area at least 10 times. Fungi increase nutrient uptake, especially phosphorus and zinc. Okay, and fungi suppress pests and diseases. And fungal networks also build aggregates. Okay, so these are very, very important. You see that plant there to your right. You can see how the root system is vastly expanded whenever you have association with mycorrhizal fungi. Okay, so plants colonized by mycorrhizal fungi, they can actually grow 10 to 20% faster than non-colonized plants, and you actually see crops mature quicker, even though they're giving away up to 40 to 50% of their photosynthate to support mycorrhizal network. Okay, one of the reasons for that apparent paradox is that plants colonized by mycorrhizae exhibit higher leaf chlorophyll content and higher rates of photosynthesis than non-colonized plants. So this enables them to fix greater quantities of carbon for transfer to fungal hyphae in that soil. And this all leads to what I'm going to show you in the next slide, which plants and mycorrhizae feed a wide range of associative bacteria, which form active colonies on the root. They can include things like nodules, but they can also include free-fixing nitrogen bacteria in and around the on the root and on the fungal hyphal tips. That once you get that established in the soil, these things are producing enzymes which solubilize otherwise plant unavailable nutrients. A lot of these fungi put out oxalic acid. So that really helps. There's a lot of beneficial interrelationships going on as we transition to a soil health management system. Okay. Now, this particular slide, this is just an example of the nitrogen fixing bacteria. The nitrogen cycle is propelled in part by specialized bacteria. One of the most important systems is maintenance of terrestrial life and living organisms. They produce vital organic compounds, the building blocks of life. You just get a lot of amino acids and nucleic acids. When doing this, they are using nitrogen, okay? And so in agricultural systems, the need to understand the nitrogen cycle is paramount, okay? If maximum use is going to be made of both fertilizer-derived nitrogen and soil's nitrogen reserves, you will learn more about this in the upcoming presentation on ecological management. So when we think about all this, this is a very important process. It's called nitrogen fixation. You get these nitrogen molecules are being taken out of the air. They're actually being fixed. They're actually being combined with oxygen and hydrogen, and they're producing ammonium. Okay, and then eventually we get the production of nitrate from those regulators, or denitrifying, I should say, bacteria that convert ammonium into nitrate in these soil systems. So it's a very important process, and it's that this nitrification, or this nitrogen fixation, I should say, okay, is accomplished from nitrogen fixing things, free-fixing nitrogen bacteria that are also in the soil on top of what you see here in this slide. but the free-fixing nitrogen bacteria are things like Azotobacter, Azospirillum, Clostridium, okay? And these are microorganisms that are in the soil. These are the ones that don't associate with the legumes, but then you also have those that associate with the legumes that form nodules on that particular plant, such as Bradyrhizobium and Rhizobium that you see here, that you see on most clovers, okay? So we don't mean to suggest that you memorize these, fancy names or anything like that, but we want you to focus on the fact that nitrogen fixation, as well as the recycling of carbon and sulfur, and all this requires these microorganisms in that soil. Okay, these are always taught as chemical processes, but they're really biological in nature, and bacteria carry out these processes in the soil, forming symbiotic, beneficial relationships with plant and, these mycorrhizal fungi that I mentioned to you earlier. And then, of course, they're feeding these free-fixing bacteria, okay, like Azotobacter and Azospirillum. They feed them carbon and photosynthate to perform their function in the soil. So it really sounds like a case of biology creating the chemistry, which it does. They fix nitrogen, but they produce nitrogenase enzyme, which gives them the ability to fix atmospheric nitrogen, okay? So we don't want you to inhibit these particular processes, so we need to think about what we're doing when we're transforming into a soil health management system. Just know that these aerobic nitrogen fixers, like Azotobacter, for instance, why they're so important, they can actually fix 10 micrograms of atmospheric nitrogen from every gram of carbohydrate that they receive from the plant, or indirectly through the mycorrhizal fungi, okay? So this is a fueling process in that case. Go to the slide here. So the next hotspot we're going to talk about for ecosystem engineers is the litter layer. This is the detritosphere. So we're moving from that rhizosphere zone to a zone above mineral soil surface known as the detritosphere, the litter layer. This is where you have those recognizable and plant animal detritus undergoing decay, like I showed you in the video. And within this particular hotspot, there are high concentrations of saprophytic fungi, mites, nematodes, and some macrofauna, beetles and centipedes. Okay, within this hotspot, there's other things such as springtails and mites. Those are tiny arthropods. They actually eat bacteria and fungi associated with decaying vegetation. Remember I told you about those trophic levels? They're all adding to the nutrient cycling processes. They're all being regulated. They're all eating other organisms. So these macrofauna help to shred. They help to mix. They help to fragment residues in that surface, and they increase that surface area for the saprophytic fungi and bacteria to release specialized enzyme to break down all of those residues. And that is where that is the particular function within that soil. Now, we do get a lot of benefits from this particular system and this litter layer. It helps us to protect the soil. It conserves soil temperature and moisture, and it also is a carbon source for all those soil microorganisms in that new habitat. So these ecosystem engineers, also assist in dispersal of even more biology, microbial spores and things within the soil system. They spread all that through that soil. They influence that microbial activity, and they really, really help us with increased nutrient cycling in the process. The next hotspot that we're going to talk about is a hotspot for ecosystem engineers. This is where you have the root earthworms and the root channels, known as that drillosphere that I mentioned to you. But it really mixes and moves residues in that soil. And you have large pores, you have nutrient richness, microbial enriched areas of the soil now because of these microorganisms. And you have air and water flow. Earthworm tunneling, if you get anywhere from 18 to 22 earthworms per foot, like I've seen it many times in corn situations, you get a lot of good air and water flow. You actually increase the infiltration rates. So roots grow and they take advantage of all that. Just know that especially earthworms, just to give you a little bit of information here, they tend to, these earthworm burrows and tunnels that you get into the soil tend to be very stable because actually their walls are lined with organic matter drawn in or secreted by those particular earthworms. And they often have a higher bulk density than that of the surrounding soil. So what it does, it actually adds a lot of stability. It really assists with water infiltration into the soil system. And if you're working with rain or dry land type of systems and you're depending on rainfall, these can be very, very important to you. Okay. Now I'm going to get into discussion about the pore sphere. This is the hotspot for chemical processors and regulators in the pore spaces. And it's created via roots, organisms, and soil health management. Has a lot to do with this. It's also known as the lungs and circulatory system of the soil. Okay, this is where you get good air and gas exchange. Okay, in this zone, air and water flow are regulated, nutrients are recycled within these systems, and transported in the soil water. And the site of mycorrhizal entanglement also, it's also sequestration of water and nutrients, and it just really serves as a biological highway, as many soil organisms can only move through water and water films. They have to have an aquatic environment. Okay, so we get good airflow, and we get good water flow and storage and availability, and we create these biological highways to get more and more biology through that soil system. So just know that plant roots, earthworms, and other arthropods, they rearrange all these soil particles. They create smooth, cylindrically shaped macropores or biopores. And as biopores can extend considerable distance into the soil, I have seen conditions where we have earthworm tunneling down to 5 and 6 feet, and I've actually seen corn roots growing in those earthworm tunnels down to 5 to 6 feet down into the soil. So these things are very, very important. It really helps increase the amount of organic content and carbon that goes down into that particular soil system. So just know that when we negate all that through mismanagement, you get poor air exchange. It leads to anaerobic condition. Organisms that can tolerate this tend to be those that cause disease, and they produce byproducts that inhibit root growth, like alcohols and things of that nature. So we have to really get this mixing of this soil, create this aggregated soil to create the diverse habitat that we need to have. Okay, so just know that that's what we're trying to strive for. We're trying to strive for a healthy soil rather than one that is more compacted. Okay, trying to support those biological highways. So now we're going to talk about the agratosphere. The agratosphere is are these aggregated surfaces that we have within the soil system. This is where get my slide to work here. This creates stability and resists erosion within the soil system. It's where we have minerals and organic materials that can be accessed. That actually increases the cation exchange capacity of a particular soil when we increase aggregate surfaces. We also protect organic matter and microbes. I just told you that we had free-fixing nitrogen bacteria, for instance, that are within aggregates within that soil system. Okay, very important. This also supports pore space, because as you create flocculation, as you create aggregates within the soil, then you have pore spaces being created in between the aggregates. So that's very, very important. And then it's also created by microbial glues, and we also remember all that fungal hyphae that I showed you that was on that corn root system, how that goes out through the soil and forms macro aggregates. Okay, so just know that lack of good soil aggregation results in compacted soils. Got to get aggregation out there. We have to restrict that. If we don't get good soil aggregation, we're going to restrict root growth. We're going to provide poor root zone aeration, poor drainage. Okay, and we're going to be lacking things like free-fixing nitrogen bacteria like azotobacter in that, because you don't have an aggregated soil. So just know that these things are very, very important, okay, to your system. And you need to understand that soil compaction is a biological problem. It's related to decreased production of polysaccharides and glomalin in that soil. And those polysaccharides allow compacted clay soils, for instance, if those are primarily our prime farmlands, to flocculate or clump up, creating needed space, when in turn it creates space for the microorganism and creates more habitat, okay? So just know that we've got to get this aggregation in the soil and prevent that soil compaction, okay? Which is due to lack of living roots and mycorrhizal fungus in that soil. So do a good job of trying to understand that when you transition to a soil health system. Now we're going to talk about soil organisms that physically stabilize soil aggregates. Plant roots, of course, and mesh soil particles. Earthworm casts also assist. Fungal and bacterial filaments physically in mesh soil particles within that particular soil system. We get a lot of stabilization of soil structure by things like actinomycetes. We also have other types of fungi that come into the soil system. As you leave residues on top of the soil surface, you create more and more habitat for different types of microorganisms to help you and assist you in this situation, okay? Soil organisms also assist us in chemically stabilizing soil aggregates. You know, they produce those polysaccharides that I told you about that allow not only the binding of soil particles and things, but also create flocculation. They allow the soil, the fake soil, for instance, to clump up, okay? And that creates pore space, that creates habitat for more forms of microbiology, okay? Soil proteins also and other biochemicals also help us to bind particles within that soil.
When we talk about what an aggregate is, Rick's got one crumbling up in his hand right now, but you know, we're going to take that larger aggregate like this and we're going to start to break it up. And all of these little pieces, as we break it up, as they kind of hold their shape in this kind of angular, smaller BB size shape, those are our aggregates. And each one of these aggregates, you know, and all of these soil particles and aggregates that are in here, these are biologically formed in these systems. So our soil microbes, in particular our bacteria and our mycorrhizal fungi and other small organisms like that, they exude biological glues that help coat those aggregates and hold them together. And so when Rick talks about building this system, he's actually building a home for the microbes so that the microbes can do their job of building aggregates. And so we really want to think about these aggregates and building aggregates in these systems.
How can the soil microbiome be manipulated in this particular situation? Just know that you need to select different plant species, varieties, or control at various plant stages. You know, things like cover crops and things like this. But you can do this through your crop rotation, crop selection, cover crop selection, planting, timing, and particular termination. Also, fertilizer, you know, when we talk about fertilization, we want to follow the 4 R's, okay? Fertilization impacts on root exudation, composition, and quantity of influences which microbes are present and how many are out there, you know. So if we practice the 4R strategy, that 4R strategy is the right source, the right time, the right placement, you know, right rate and placement of these particular nutrients in that soil system. Also, soil amendments are very important. Also, these can include biological that help. You know, many times you can stimulate beneficial microbes around the soil system to defend against plant diseases and pathogens. Go into a little bit more detail in the next slide. The soil biologicals or biological amendments, they have promise and consistency. We just have to learn how to use them. And then also, we want to manage the environment to minimize stress. You know, pathogens, drought, temperature extreme. We can do this by following the 4 soil health principles you know, that were mentioned earlier. And that gives us the ability to kind of regulate temperature, moisture, and we maximize the presence and duration of the particular hotspots.
A biological hotspot is a location or a region within our soil profile where we know that if we're going to look for soil organisms, that is a place where we will more than likely find them. If we think about, say, a cornfield, right? We have, in most typical scenarios, besides Rick, because he's not typical, he is well above average, but in most typical scenarios, we got 30-inch rows. So that area in between those rows, that's kind of a dead zone because there's not a lot of vegetation there. There's nothing covering the soil surface in conventional systems. And so we want to think about those hot spots where if we were to look for soil organisms, we want those hot spots to be the areas where we would typically find them and have greater success of finding them.
You can kind of think of it too as kind of an interface between that ecosystem underground and what's happening above ground. So it's a linkage between all that ecosystem underground and what you see above ground.
So what we see here, this is a little bit of a discussion that we're winding up here about below ground competition. I'm going to bring all this up for now. But what you see here in the middle is or just to start up in the upper left, you see a nematode trapping fungi. These are known as trichoderma. They produce cellulase, they break down plant material, and they produce chitinase also, which means they can break down chitin, or in other words, they can attack insects, nematodes, or other fungi, okay, because they have chitin. They're made of chitin. So trichoderma come into the system when you no-till and add cover crops to a rotation. You leave residues on the ground that provides a food source for them. And then those residues and then the biology that comes in there can also be a resource, food source for those particular forms of biology that you're bringing into that soil system. On the lower left here, you see what are considered to be vampirillid's. These are actually protozoa that are eating a fungal root pathogen known as take-all disease in wheat. So you can see that they're eating little holes into those hyphae that are there. Okay, so that's a form of biological control. We also have, within these particular soil systems, one of the things that we have to contend with is that you see here in the middle is that springtails or columbella or springtails can help us out tremendously with things such as Rhizoctonia solani. That's a seedling disease. And I particularly pay attention to that here in Texas in the South because that helps us present disease or seedling disease known as damping off on cotton. Okay, so if we have these beneficial microorganisms like springtails, they actually consume this particular fungal disease or pathogen. Okay, and that's what you see here in the middle. You see on the left roots with springtails that are very healthy, and then roots without springtails on the right. Okay, so this is very, very important to understand. Also, I do know this, and this does apply to y'all, but we also have plant growth promoting bacteria, for instance, that produce things like antibiotics. One of them is Pseudomonas fluorescens. It produces pyrrolnitrin, which is able to prevent damage of Rhizoctonia solani also, okay, during damping off. And so these help us with seedling diseases and stuff like that. So we have to kind of reverse our program and go back to producing these biological systems. I also know that things like Pseudomonas are very important to you. Just to wrap up, they're very important to you when it comes to manganese acquisition in the soil. You need that in order for plants to convert glucose to sucrose. So these are very beneficial microorganisms within that soil system. Okay, to summarize, managing for soil biology, most ag soils are carbon depleted. Disturbances destroy habitat and hyphal networks like mycorrhizae. Bare fallow fields provide little protection. There's no carbon going into the soil system, no live plants putting carbon in the soil. Agrochemicals have mixed effects. Many fertilizer concentrations also are too high for symbiosis, like I'm going to show you in the next slide. But we want to manage for those hotspots, support biology, build aggregates, create pore space, protect the habitats that are created, feed the soil so it can feed us. And we want to optimize biological nutrient cycling and optimize plant microbe interactions for plant defense and optimization. We don't want to do anything that inhibits that particular process. And I'm going to show you something here, about understanding about fertilizer use and it having the ability to inhibit certain types of biological activity. What you see here, this is where nitrogen has been placed below the seed, and you see how the plant has started to associate with biological activity on top. You see how the soil is sticking to the particular roots. They're forming rhizosheaths. But as soon as that particular plant root gets into the fertilizer, the inorganic fertilizer, it inhibits the formation of those rhizosheaths on those roots. So just note that healthy rhizosheaths above the seed, and then you see how we have clean white roots below, where we're not getting the biological activity and not getting the root exudates put out into the soil. So one of the things that we want to do is just recognize that. We want to make sure that we feed the below ground life. The way that we do that, we choose practices that provide diverse, near continuous inputs and build reserves. How can we provide and protect that habitat? Choose those practices that help minimize that disturbance, help us to build aggregates in the soil rather than deplete them or degrade them. And we want to provide those food sources, both soil organic matter, organic carbon, and those residues that are needed for that biological habitat to be there. Choose practices that support that stable habitat from major swings in temperature, water, and chemistry. Okay, so we want to make this a constant particular process within our soil system. And then we also want to understand, as we choose those particular practices within these particular soil systems, we want to make sure that we optimize function, okay? And essentially what that means is that we're going to start out focusing on that rhizosphere that I told you about. We're going to create that litter layer, okay? We want to make sure that we get those added root, and worm channels in that soil. We want to get that aggregated surface started that really helps us improve cation exchange, nutrient cycling, et cetera, pore space. And then we also, as we get that particular pore space, we create these particular conditions. That's going to be very beneficial to establishing all of these biological hotspots that I mentioned to you. Just know that 20% or more of that carbon that's fixed by plants is released out of those roots, and it's fed through root exudates to these soil microorganisms. This is not a leaky system, but rather it's evolved over time to understand that it stimulates the microbial activity in that soil, okay? And the bacteria and fungi taking up these compounds in return aid that plant in many, many ways. And one of the key things is that it's going to support your next presentation when you start to understand that you're trying to protect the soil aggregates and organic matter, because you're trying to minimize disturbance, maximize soil cover, and then you're trying to feed and fuel that soil biology by maximizing biodiversity and maximizing continuous living roots.
If we do not eliminate tillage, okay, and people constantly are tilling, all they're doing is breaking these systems down, then the mycorrhizal fungi are rebuilding that system just in time to have it wiped out again by another passive tillage. If that just keeps occurring, there's no soil health being built, because all they're doing is trying to rebuild their communities.
Okay, so this is what we're trying to do within these systems. We want to be following these basic soil health principles to help us manage soil biology in that soil, maintain soil organic matter, aggregates, reduce erosion and runoff, buffer those temperatures that are there, reduce evaporation, feed and help to break those disease cycles by feeding plant beneficial microorganisms in that soil, increasing that soil organic matter, increasing the nutrient cycling processes, enhancing growth. Because of all of that, and increasing these predator and pollinator populations, all these different things that can help us, many things that lead to the improvement of soil function. Okay, that concludes my presentation, and somewhat brief when it comes to the discussion of soil biology, but we can look towards maybe some questions and answers coming up.
You talked about different root systems and stuff like that for diversity. Like, would you mean like if a guy has wheat in a rotation, that instead of planting wheat so often do barley or rye? Would that make a difference with the root systems?
I think most of it is this discussion to understand, some of these plants, all of those have fibrous root systems, and probably your cereal rye, some of these other things will probably go deeper into the soil profile than some of your other particular crops. It just, depends. But a lot of times I'm also talking about diversity in your cover crop systems that you put in between cash crops. And in those types of covers, we really need to have that biodiversity where you have both fibrous and tap-rooted systems. You also have the biodiversity where you have legumes and non-legumes, so that you get all the particular nitrogen fixing types of bacteria working for you. Not only those that are legumes, but also those that are free fixing nitrogen, those that are solubilizing phosphorus. And you've really got to get that root system throughout that soil system. So having both tap roots and fibrous roots all benefits grasses and broadleaves within that system. Also understanding warm season and cool season, it all adds to those different things, and different crops in your rotations mean different root systems, and as to the depth and the amount of biological activity that are associated with those certain types of plant species.
One of the things about the soil biology module, I find that one very interesting because I kind of feel like it's really the basis of everything. One of the things I find interesting about it is how much we still don't know about what's going on with all those interactions and everything. I guess my question was kind of who is on the kind of the leading cutting edge of, doing that research and finding out how all these things are interacting within the soil between these organisms. Is there one group that's doing that, or? There's
several, I guess you could call them soil health innovators, or people that I use as mentors. I've learned a lot from Jay Fuhrer and John Stick up in North Dakota, but I also have other mentors like Dr. Elaine Ingham. Okay, she's responsible for our soil food web, that we have, so I've learned a lot from her. Also, another lady by the name of Dr. Christine Jones. I've learned a lot about free-fixing nitrogen bacteria and some of the other associated bacteria that really have to be there in natural systems and their associations with mycorrhizal fungi. Dr. Amaranthus, who's now, I don't think he's in research anymore, but he's sold his company, but he's very prominent in mycorrhizal fungi understanding. We know we can pick up amino acids and things like that because of these associations. So these are all added types of functions when we add this different habitat, this different microbiology activity. But there's a lot of people out there that help us, with this understanding. Thanks, guys. Take care. Be sure to contact me if you need anything.